Hello, everyone, and thank you for joining the H.C. Wainwright 2021 Global Life Sciences Conference. My name is Rosario Graziano, and I'm an associate here at the firm. While we are all virtual this year, we are confident we will be able to provide value to with over 425 companies presenting at this conference, as well as via your interactions through one-on-one meetings. H.C. Wainwright is a full-service investment bank dedicated to providing corporate finance, strategic advisory, and related services to both public and private companies across multiple sectors and regions. We have a total of 18 publishing senior analysts and 493 companies covered across all sectors. Please visit hcwco.com for more information. From a logistics standpoint today, please make sure to reference your online conference portal that provides your individual links to all meetings and presentations. Panels and presentations are live and on demand from March 9th to the 10th. With that said, we hope you have a productive and enjoyable day, and I would like to introduce our presenter today. Please welcome Per Norlin, the CEO of Alligator Biosciences. Per?
Thank you so much. So let me introduce you to Alligator. Alligator is a public immuno-oncology company, and we are differentiated through the development of tumor-selective immunotherapies. This is essentially therapeutic antibodies that activates the immune system strongly in the tumors, but not elsewhere in the body. And I will start with an overview. Next slide. And the next slide again. So we have two lead products, two clinical products, ATOR-1017 and mitazalimab or MITA, and they both have enormous potential. They are moving now into clinical phase 2. We have preclinical and clinical data that supports a leading property within their respective fields. We also have a strong innovation track record within the company. We have brought 5 clinical stage products all the way from the ID to clinical stage. And that is using our internal technology platform. And we have another new drug concept, which was launched recently, NeoX Prime. And this is a more patient-specific immunotherapy. I will come back to that at the end of the presentation. And we have a validated business model. Let's take a look at that on the next slide. Our focus is immuno-oncology, and it's a very effective therapy, but it's only effective in about 1 out of 5 patients. Our goal, of course, is to increase that, and there's an international effort to increase that proportion to higher than 1 out of 5. We have leading candidates within 2 of the key pathways for this effort. And they are both moving towards phase 2, and that is a very important value inflection point. On a positive phase 2 trial, generally valuation increases in the range of 5-fold. On the business model, we develop to clinical stage and base our revenues on royalties and milestones upon out-licensing. And we have a track record. We have generated more than $50 million over the last 5 years in such milestones and upfront payments. And we intend to do it again. ATOR-1017 and mitazalimab being some of the candidates. So let's move to ATOR-1017. Next slide. ATOR-1017 is a 4-1BB antibody and is developed to increase the number of patients that responds to immunotherapy. The properties of the product, it's a tumor-selective 4-1BB antibody, and it has a lot of potential for synergy with PD-1 products. As you know, PD-1 releases the brakes, whereas 4-1BB pushes the gas, and both are needed to pick up the speed significantly. There's also emerging clinical validation for the targets, and that, of course, increases the chance of success. 1017 or ATOR-1017 is in late phase 1, and we are planning to present data towards the summer. So what about the differentiation of this product? Next slide. Here is the first generation products. On the left-hand side, we have the BMS product, urelumab. This is a strong antibody, but also quite toxic. And that's because of general activation of the immune system. And that resulted in a very low tolerated dose in the range of 8 mg. On the right-hand side, we have the Pfizer product, utomilumab. They sort of overcompensated to improve safety. So they have a weak product, and it's only activated under perfect conditions. And as you can see, the tolerated dose is about 100-fold higher. 1017 then. Here we have a strong activating antibody, but it's tumor selective. And that means we can get both strong efficacy and manageable safety. That's supported by our preclinical data, but also by emerging clinical data. So first, how did we go about to make it more tumor selective? Next slide. Here we have two graphs trying to show this. The antibody requires cross-linking of two cells in order to have full effect. If you look on the left-hand side, and that's a circulation, it can bind to the target 4-1BB, but it cannot bind to the other receptor, the IgG binding Fc gamma receptors. That's because there is tremendous competition from circulating IgG in the circulation. So there will be no effect in the circulation. In the tumors, on the right-hand side, both receptors are available, both 4-1BB on the T cells and Fc gamma receptors on other immune cells. And there you can get strong immune activation. And we have preclinical data to support exact this theory. Next slide. On the left-hand side, we can show very strong activation in the tumor area, and that's seen in the green bar. And on the right-hand side, you can see there is no activation whatsoever in the circulation. And that's exactly what we want to show, a tumor-selective immune attack. And this is what we call the second generation 4-1BB immunotherapies. And what about the competitors? Next slide. See if I can change the next slide. So here we have 4 different products. You can see the ones with the same structure. 1017 has an IgD4 structure, which we believe is ideal to allow tumor selective activation. And that is shared with 2 other products, Adagene and Compass. We also have a ligand blocking property, and that's a fact that improves safety. That's something we share with Adagene, but not with Compass. We have selected a binding domain that allows higher efficacy, and that's also illustrated in the preclinical data. So we have a very high potency, and all in all, it seems we have a best-in-class potential with this product. And we are in late stage phase 1. So what about the clinical data? Next slide. Dose escalation is ongoing. We have cleared a dose today that is 100 milligram, which is about 10 times higher than where the first generation benchmark product urelumab had to stop. Still, adverse effects are only low grade, and there are signs of immune activation. We will have a readout of this study in Q2, and we will present safety, pharmacokinetics, potential efficacy, and also recommended phase 2 dose. And the intention here is to start clinical phase 2 in gastric cancer during the autumn, with potential for first efficacy data in the second half of 2022. Next slide. So let's move on to mitazalimab. Here is a different approach to increase the number of patients that respond to immunotherapy. Mitazalimab is a CD40 agonist, and it has potential to have efficacy in PD-1 refractory tumors of patients, that even in cold tumors where there are very few T cells. So what CD40 does is improve antigen presentation. That in turn leads to T cell infiltration in tumors. And once T cells have infiltrated the tumors, there is potential to respond to PD-1 therapy. Phase 1 has been completed. We have more than 100 patients assessed in clinical phase 1, and we have submitted a CTA to start clinical phase 2 in pancreatic cancer. What about the competition? Next slide. Again, we have a product which is results in tumor selective immune activation, and that's again based on the requirement for cross-linking with Fc gamma receptors. There are 4 such second-generation products, and on these, mitazalimab or Mita is the only one that can be dosed at therapeutic range dose levels, which is in the range of 1 mg per kg. Let's look at the details on the next slide. So the 4 second-generation products are shown in green, and as we see mitazalimab can be dosed at a dose that allows superior efficacy of this product if you compare to and that's highlighted here. From clinical phase 1, we have the strongest data presented so far. We have 10 patients with stable disease and 1 partial responder. In phase 2, there is only 1 product so far that has presented data of these 4 second-generation products, and that product has shown some promising activity in pancreatic cancer. Let's look at the next slide. So CD40 has emerging clinical validation in pancreatic cancer. If we look on the right-hand side, the black bar is showing the response rates of current chemotherapy treatment. The red bar, on the other hand, is the addition of PD-1 on top of chemotherapy. And as you can see, there is no additional effect of PD-1. The explanation is quite simple. This is an extremely cold tumor, and therefore there are very few T cells in the tumors. So PD-1 has no effect because there is no T cells to activate. But then look on the right-hand side, on the green bar. When you add CD40, CD40 will increase T cell infiltration into the tumors, and that by itself seems to increase response rates, in this case in the range of 40%. And the gray bar then shows, and this is exactly according to theory, that the increased T cell infiltration also allows for effect by PD-1. And as you can see, the gray bar, the combination of CD40 plus PD-1 on top of chemo gives a very impressive response rate in this study. So we are also planning now a study, a phase 2 study in pancreatic cancer. It's slightly different, so let's look at the details. Next slide. We will combine with standard of care chemo, in this case, modified FOLFIRINOX. In the previous slide, it was with gemcitabine. And the aim, of course, is to add on PD-1 once we have shown a signal in combination with chemo with our mitazalimab. So let's look on the right, which is the design. And it builds on the differentiated properties of this antibody. It is, as I said, the only product that can be dosed at therapeutic range dose levels. And that is because it has a better tolerability. That also allows for combination with a more toxic chemotherapy like FOLFIRINOX. But the advantage is FOLFIRINOX is a stronger chemotherapy cocktail and also will result in release of more tumor antigens. In addition, since it's slightly more toxic, it's usually used for patients with a bit better quality, with a better status, and that basically means that they probably have a better immune system in place. So we combine with chemo, and the chemo is given at day 0. That will result in release of tumor antigen. 2 days later, mitazalimab will be administered, and then that will activate the dendritic cells that have taken up tumor antigens and will present that effectively to the immune system. And then this sequence of events will be repeated continuously throughout the study. We aim to start in Q2. The first handful of patients may be communicated at the end of this year in Q4, and we hope for a larger readout in the second half of 2022. Apart from these 2 clinical products, ATOR-1017 and mitazalimab, we also have a novel drug concept developed, Neo-X Prime. So let's look at the next slide. Again, the main issue today is that only 1 out of 5 patients responds to chemotherapy. And it mainly has to do with the failure of the immune system to identify what to attack in the patient, to identify the patient-specific tumor antigens. Neo-X Prime solves this by bringing these patient-specific neoantigens to the dendritic cells and making them selectively present those neoantigens. And that makes it a more personalized immunotherapy treatment. If we look on the lower left, this is how it works. We use a bispecific antibody that on the one end can bind to circulating tumor exosomes, and then the other part can bind to the antigen-presenting dendritic cells. So the bispecific antibody can catch the tumor exosomes in the circulation, bring those to dendritic cells, and make sure they selectively present the neoantigens of that patient at that point in time. So it sounds good in theory, but what's really is interesting is the data we have generated. This new X prime concept outperforms anything we previously have been tested in our models. If we look on the right-hand side, this is a comparison. So at the blue bar, we have two antibodies in combination, and on the right-hand side of that, the green bar, that is the same antibodies that has been connected to each other in the Neo-X prime format. And as you can see, there is a tremendous increase in efficacy. So very promising, and we are today looking for partners to develop those products and bring them through preclinical development and to clinical assessment. So I would like to summarize this presentation in the next slide. We have two clinical assets today that are approaching clinical phase 2. That's ATOR-1017 and mitazalimab. ATOR-1017 is in late stage phase 1. We have promising clinical data, and we will present that in Q2, the data including pharmacokinetics and potential efficacy, and a recommended phase 2 dose, and then submit a CTA for clinical phase 2 in gastric cancer in the second half of this year. The other product, mitazalimab, will start dosing in clinical phase 2 in Q2, assuming that we get our CTA approved. It has been submitted in pancreatic cancer, and we hope to present the first few patients in this phase 2 efficacy study by the end of this year. So thank you so much for listening.
Well, thank you, Per, for your informative presentation on Alligator Biosciences. I also want to take a second and thank all of our presenters for taking part in what's been a very productive and informative series of presentations. We appreciate the time and effort that went into preparing them, and hopefully our next conference will be one that can be held in person rather than virtually. In the meantime, we are very grateful for your flexibility and your presence online this year. Thank you again from the H.C. Wainwright team.